Today we will talk about enhancing tumor immunogenicity using immunogenic cell death induction. The immune checkpoint inhibitors help the patient immune system to recognize and attack cancer cells. Therefore, the immunogenicity of cancer cells is a fundamental determinant of immune checkpoint inhibitor response. So if the tumor has no or very low immunogenicity, this will not respond to immune checkpoint inhibitor. And in this case, we have to use some immunogenic drugs, for example, to induce immunogenic cell death. This will enhance the tumor immunogenicity in order to convert the tumor into immunotherapy responsive tumor. So what is the definition of tumor immunogenicity? Here, many DNA mutations cause the cancer cell to make mutated proteins, or what we call new antigens. And the extent to which cancer cells make the mutated proteins or new antigens is called tumor mutational burden. So tumor cells commonly express two types of antigens. Those new antigens that are mutated proteins and different from any other normal proteins in the host, or tumor-associated antigens that expressed in unusual tissue or significantly high amounts. The tumor immunogenicity is usually determined by two independent factors, but both are required. Number one, the antigenicity of tumor cells, the extent of expression of the new antigens and the tumor-associated antigens. Number two, the processing and presentation of tumor cells, of tumor antigens by dendritic cells. So here, after formation of the mutated protein, it will be expressed by the dendritic cell as major histocompatibility complex, and the dendritic cells will migrate to the tumor-derived lymph node for priming of the T cells. Then T cells will be infiltrating the tumor and recognizing this new antigen expressing a cancer cell and attack this cancer cell, leading to their elimination. So tumor with high tumor mutational burden should respond well to immunotherapy. And for a new antigen to be immunogenic, it must be processed and presented by the major histocompatibility complex molecules on antigen-presenting cell like dendritic cells. So after uptake of antigen by dendritic cell, it will process this antigen in the form of peptide, major histocompatibility complex, and will prime the T cells in the lymph node. These primed T cells will migrate into the tumor and recognize this antigen-expressing cancer cells, leading to their elimination. However, only 1% of mutated proteins expressed in tumor is immunogenic in cancer patients. And this low immunogenicity can be due to many factors. For example, can be due to molecular defects that prevent cancer cells from releasing damage-associated molecular patterns, or DAMPs. So many patients present intrinsic resistance to immunotherapy, and this resistance can be explained by different factors. Number one, loss of antigenicity in tumor cells. So the resistance can be due to loss of immunogenicity or antigenicity in tumor cells, and this can be due to a low level of new antigen expressed, or disappearance or impairment of the antigen presenting capacity of dendritic cells, or inability of cells to respond to some stimulating factors like interferons, for example. Also, the resistance to immunotherapy can be due to absence or inhibition of danger signals linked to inflammatory and immune response in the tumor microenvironment, or due to enrichment of immunosuppressive cells in the tumor microenvironment, for example, T regulatory cells that can block the infiltration and cytotoxic function of CD8 positive cytotoxic T lymphocytes. So how to enhance the tumor immunogenicity? There are different immunoengineering approaches to enhance tumor immunogenicity. Today, we will talk about immunogenic cell death induction. Also, cancer vaccines can be used to deliver some antigens to dendritic cells to initiate the anti-tumor immune response. Other approaches also can be used to enhance the expression of tumor-associated antigen on the cell surface, like, for example, epigenetic drugs. So the first strategy to enhance the tumor immunogenicity is immunogenic cell death induction. The definition of immunogenic cell death induction, also termed in situ cancer vaccination, it's a type of cell death. What is the difference between immunogenic cell death and normal or regulatory cell death? In immunogenic cell death, there is release of what's called DAMs, or damage-associated molecular patterns. This can induce inflammatory response to activate the innate immune response in the form of dendritic cell recruitment and activation, and will lead ultimately to adaptive immune response by priming and activation of T cells. So what is happening here? The dying cancer cells, after treated with immunogenic cell death inducer, are subjected to cellular stress. This cellular stress will cause the cancer cells to release some danger signals, or what we call damage-associated molecular patterns, or DAMs. In immunogenic cell death, these dying cancer cells release DAMs, including release of ATP, which will induce activation and recruitment of dendritic cells, release of high mobility group box1 protein, HMGB1, release of interferon 1, release of some heat shock proteins, and also, more importantly, translocation and surface exposure of the endoplasmic reticulum protein, calreticulin, CRT, from the endoplasmic reticulum will be surface exposed on the surface of dying cancer cells. So the released DAMs, together with CRT, calreticulin, will interact with complementary receptors on the dendritic cells, leading to their activation, maturation. This will enhance the antigen presentation capacity of dendritic cell, will induce their migration to lymph node to prime the T cells and convert the prime naive T cells into effector T cells. Now the prime T cells will be able to infiltrate the tumor and release some interferon gamma and stimulatory factors, and will attack the cancer cells, leading to their elimination. So the molecular mechanism of immunogenic cell death include interaction of the released DAMs with complementary receptors on the dendritic cells, leading to their activation maturation. It starts with release of ATP. The ATP will induce recruitment and activation of dendritic cells by binding to some purinergic receptors on the surface of dendritic or antigen-presenting cells. Also, the surface-exposed protein, calreticulin, will bind to complementary lipoprotein receptor-related protein 1, or what's called CD91, on the antigen-presenting cells to enhance the phagocytosis of dying tumor cells by dendritic cells and enhance antigen uptake. Also, the released HMGB1 from the nucleus to the extracellular space will bind to toll-like receptor 4 agonist and advanced glycosylation end product specific receptor, leading to maturation of dendritic cells and enhancing its antigen presenting capacity. Finally, the type 1 interferons released from the immunogenically dying tumor cells will have immunostimulatory effect to the antigen presenting cells via binding to their complementary receptors. According to Kroemer-Zitvogel model of immunogenic cell death, this process is a multi-step process, including different steps. Number one, release of find me signals, which is mainly ATP to attract the phagocytes or dendritic cells. This is the recruitment phase. Then really expression of eat me or digest signals, mainly calreticulin and phosphatidylserine. This will facilitate the recognition of dendritic cells by recognition of cancer cells by phagocytes or dendritic cells. Also, release of danger signals, mainly high mobility group box1 protein, HMGB1. This will enable the cancer cells to stimulate powerful anti-cancer immune response by activation of antigen-presenting cells. All this in addition to the capacity of caspases to cleave and release the apoptotic cell-associated antigenic fragments. Those fragments of antigens will be transported from phagosomes to cytosolic proteasomes in the form of peptides, which will then transferred into the lumen of endoplasmic reticulum and bind the appropriate MHC class 1 molecule. Now, dendritic cells will be able to induce priming of T cells, and T cells will infiltrate the tumor and attack the cancer cells expressing those antigens. Only few agents have the ability to elicit anti-tumor immunity by inducing immunogenic cell death of tumor cells. For example, different strategies can be used for induction of immunogenic cell death. Some anthracycline chemotherapeutics like doxorubicin and metoxantrone, oxaliplatin, some targeted therapeutic molecules like bortezomib, also radiotherapy, photodynamic therapy, photothermal therapy, and oncolytic viruses can be used to induce immunogenic cell death of cancer cells. In all these strategies depend on generation of reactive oxygen species. This is very important to induce stress to endoplasmic reticulum, oxidative stress to endoplasmic reticulum, resulting in release of DAMPs from the immunogenically dying cancer cells. We have two types of immunogenic cell death inducers, type 1 and type 2. Type 1 act mainly on the focused sites, nucleus, cytosol, or plasma membrane, to induce the cell death by different mechanisms. For example, those chemotherapeutics can act on the nucleus by targeting proteins involved in DNA replication and repair, or can act on targeting certain cytosolic proteins, the cytosol, or targeting certain transmembrane proteins or channels in the plasma membrane. This to induce cell death. However, also type 1 immunogenic cell death inducers act by secondary or collateral effect to induce endoplasmic reticulum stress, to induce release of DAMPs. But their main effect is mediated by the nucleus or cytosol or plasma membrane targeting to induce cell death. Its secondary effect is mediated by acting on the endoplasmic reticulum through secondary or collateral effect to induce release of DAMPs. However, type 2 immunogenic cell death inducers, like the photodynamic therapeutic molecule like hypericin, for example, those act only on the endoplasmic reticulum as a focused or primary site, and their action on endoplasmic reticulum to induce stress mediated by mainly reactive oxygen species generation. This endoplasmic reticulum oxidative stress will result in both apoptosis and release of DAMPs. The advantage of immunogenic cell death inducers compared to other vaccines. Usually, conventional vaccines used for cancer use only selected tumor-associated antigens. So they fail to reflect the multitude of tumor antigens that may evolve during immune editing by the tumor. They usually use nanoparticle to deliver only one or two antigens to the dendritic cell to initiate the tumor response. So they will lack the response to other antigens released by cancer cell. However, in immunogenic cell death, we use the whole tumor-related antigenic repertoire. That means the immunogenic cell death will activate the antigen-presenting cells and help them to present the full complement of tumor-associated antigens, mutagenic and non-immunogenic. Also, the whole cell cancer vaccines can be used by using the tumor cell lysate prepared by freeze-thawing or irradiation of cancer cells. However, this technology usually suffers from low activation efficacy of dendritic cells. So it's another type of cancer vaccines. They use the whole cancer cell by inducing tumor cell lysate formation. This can be done by freeze-thawing or irradiation. But when we use those whole cell cancer vaccine, we usually have limitation of impaired activity of dendritic cell. In contrast, in our immunogenic cell death, the major mechanism is to enhance the activity of dendritic cells, and this can be achieved through the inflammatory response mediated by release of DAMPs and ended with the priming of T cell and generation of T cell anti-tumor immune response. Now, the incorporation of immunogenic cell death inducers into nanomedicine can help increase the efficacy of immunogenic cell death induction. Because usually the conventional immunogenic cell death inducer suffers from limitations, including undesirable drug delivery efficiency to the tumor, off-target toxicity, also limited efficacy. Usually the effect of those conventional ICD inducers is relatively weak and transient and fail to protect against tumor recurrence. So incorporation of ICD inducers into nanocarriers should at least ensure enhancing the efficacy of immunogenic cell death by improving the stability to prevent premature drug release into the systemic circulation and ensure release of this immunogenic cell death inducing drug only into the tumor. Also, the nanoparticle should ensure deep tissue penetration to reach all the cancer cells in the core of the tumor. Also, we should use minimal amounts of excipients that will help the scale-up and the clinical translation potential. So we have different types of nanomedicines can be used to enhance the efficacy of immunogenic cell death. This include chemotherapy, targeted therapy, photodynamic therapy, photothermal, magnetic, radiotherapy, and finally, immunogenic cell death whole tumor cell vaccines. First, we have some of chemotherapeutic drugs, for example, doxorubicin or oxaliplatin, that can be used not only for their cytotoxic effect, but also to elicit anti-tumor immune response through inducing immunogenic cell death. However, usually those compounds have some challenges like inefficient drug accumulation to the tumor, resulting in both systemic toxicity and toxicity to immune system. So incorporating those nanocarriers in those immunogenic cell death inducer into nanocarriers will increase the drug concentration to the tumor and reduce its systemic toxicity, enhancing its immunogenic cell death inducing efficacy. But usually the disadvantage of immunogenic cell death chemotherapeutic that they induce immunogenic cell death through collateral or secondary effect on the endoplasmic reticulum. It's usually more better to induce immunogenic cell death by direct generation of reactive oxygen species and inducing oxidative stress to endoplasmic reticulum. This is usually more effective than working secondarily or collaterally on endoplasmic reticulum. In this example, this research study, they prepared core shell nanoparticles for co-delivery of two drugs, oxaliplatin chemotherapeutic and another drug called dihydroartemisinin to induce immunogenic cell death. Here, oxaliplatin acts mainly by inducing DNA damage and secondarily by generation of reactive oxygen species. Dihydroartemisinin, usually it contains endoperoxide moiety. This endoperoxide moiety reacts with the ferrous iron to generate reactive oxygen species and induce oxidative stress to endoplasmic reticulum. And this will synergistically with oxaliplatin enhance the efficacy of immunogenic cell death and promote the infiltration of T cells into the tumor. So from immunoengineering point of view, to prepare these nanoparticles, the core was composed by coordination polymerization between zinc ions and the phosphate groups of oxaliplatin to entrap the oxaliplatin into the lipid core of the nanoparticle. While the shell is composed by pegylated phospholipid bilayer containing conjugate of dihydroartemisinin with cholesterol. They are coupled with a chemical bond like disulfide bond. This disulfide bond is specifically cleaved by the high concentration of glutathione in the tumor cells. This will ensure site-specific release of the drug into the tumor, not into the circulation. And after release of the two drugs, they will act synergistically, like we illustrated, to induce immunogenic cell death and release of DAMPs that will activate dendritic cells Dendritic cells, when matured, will induce priming of the naive T cells into effector T cells that will infiltrate into the tumor. In combination with anti-PD-L1 immune checkpoint inhibitor will enhance the anti-tumor efficacy. Another type of immunogenic cell death nanomedicine using photodynamic therapy. Photodynamic therapy is a type of minimally invasive therapy of cancer that depends on two steps. Number one, we have to administer compound called photosensitizer, like hypericin, for example. This photosensitizer has the ability to be activated using light irradiation with a specific wavelengths. When activated, the photosensitizer in presence of the surrounding oxygen in the tumor microenvironment will generate highly cytotoxic ROS, reactive oxygen species. And this is the major player which will induce endoplasmic reticulum oxidative stress and inducing release of DAMPs and immunogenic cell death. Usually, the limitation of photodynamic therapy is the localized therapy because the light penetration is usually limited to the superficial tumors. Also, those photosensitizer molecules are usually hydrophobic, water-insoluble molecules, and then tend to aggregate in the aqueous media. This will reduce its photophysical activity, will reduce the formation of reactive oxygen species, will reduce its solubility, will reduce its tumor localization. And therefore, incorporation of the photosensitizers into nanoparticles can ensure its specific delivery into the tumor and reduce its systemic toxicity and enhance its immunogenic cell death-inducing efficacy. In this research study, a type of chimeric peptide nanoparticles were synthesized by coupling a photosensitizer molecule, protoporphyrin 9, with another molecule, indole diamine oxygenase inhibitor, 1-methyltryptophan. The nanoparticles were accumulated into the tumor area through the enhanced permeation retention effect. Upon light irradiation, the release of the photosensitizer will act by converting the surrounding oxygen into reactive oxygen species to induce endoplasmic reticulum oxidative stress and release of DAMPs. In parallel, methyltryptophan will inhibit indolamine dioxygenase enzyme to induce activation of cytotoxic CD8 positive T cells and inhibit the immunosuppressive cells. So both synergistically will enhance the anti-tumor efficacy. A third type of ICD nanomedicine acts by photothermal therapy. Here we have some light-absorbing agents, usually inorganic nanoparticles like gold or palladium nanoparticles. Those agents have the power to absorb light and convert the photo energy of light into thermal energy. And this thermal energy usually kills the cancer cells by disrupting the cell membrane, denaturing the proteins, and inducing DNA damage. Recently, the photothermal therapy was found to also induce anti-tumor immune response by causing the dying cancer cells to release tumor-associated antigens and DAMPs, which will elicit anti-tumor immune response. And again, the photothermal therapy-based cancer immunotherapy is limited by the reduced penetration power of laser and insufficient activation of immunogenic cell death. Therefore, we try to incorporate these molecules into nanoparticles to enhance the efficacy to induce immunogenic cell death. For example, this combination nanomedicine was developed for co-delivery of doxorubicin as a type 1 immunogenic cell death inducer and photothermal agent palladium nanoparticles. And those co-encapsulated into type of triglycerol monostearate lipid nanoparticle. Inside the tumor, the overexpressed matrix metalloproteinases and esterases will hydrolyze the lipid and release both drugs, doxorubicin and palladium nanoparticles, to kill the cancer cells through chemotherapy and hyperthermia. So after laser irradiation, the photoabsorbing agent, palladium nanoparticles, will absorb light and will convert the photo energy into thermal energy. So the temperature of the tumor has increase to 50 degrees Celsius only in 6 minutes, and this will lead to tumor apoptosis and release of DAMs. In combination with anti-PD-L1 immune checkpoint inhibitor, this enhances the immunogenic cell death and release of DAMs, ATP, HMGB1, and calreticulin, and this will promote the dendritic cell maturation and the T lymphocyte infiltration. Also, we can use magnetic nanoparticles to induce immunogenic cell death. So ferroptosis has been widely known as iron and reactive oxygen species-dependent form of cell death. Also, this ferroptosis can induce unfolded protein response, and this unfolded protein can contribute to several endoplasmic reticulum stress-mediated signaling pathways. So here, for example, oxaliplatin was incorporated into magnetic nanoparticles to promote the ROS generation effect of the released oxaliplatin, and this will enforce or enhance the efficacy of immunogenic cell death. So the internalized oxaliplatin can act by producing intracellular hydrogen peroxides through the enzymes in the upregulated into the tumor microenvironment. Then the ferric ions through Fenton's reaction will use this hydrogen peroxide to generate reactive oxygen species to induce endoplasmic reticulum stress and release of DAMs. Also, radiotherapy can be used to enhance immunogenic cell death. So radiotherapy is a type of therapy mainly acts by destroying DNA double strands. However, recently it was found that radiation treatment can also induce immunogenic cell death by triggering release of heat shock proteins HMGB1 surface exposure of calreticulin, leading to dendritic cell maturation and inducing interferon gamma producing T cells, both in vitro and in vivo. However, not all radiation treatment-induced modification can result in immune activation. For example, in some studies, they noticed accumulation of pro-tumorigenic M2 macrophages and T regulatory cells in the hypoxic areas of irradiated tumors, because we know that hypoxia can lead to resistance to radiation therapy. So to enhance the effect of radiotherapy to induce immunogenic cell death, polylactic coglycolic acid nanoparticles were prepared to encapsulate the enzyme catalase together with the toll-like receptor 7 agonist, imiquimod, as an adjuvant to increase the activity of dendritic cells. Catalase, this enzyme acts by decomposing the generated hydrogen peroxides into oxygen, and this will reduce the hypoxia and reduce the resistance to radiation. So after radiation of the tumor with X-ray, together with these nanoparticles, the effect of tumor killing was enhanced due to both catalase triggered tumor hypoxia relief, and this will reduce the resistance to radiotherapy, and also the presence of imiquimod as a toll-like receptor agonist will reverse the immunosuppressive tumor microenvironment and activate dendritic cells and cytotoxic T lymphocytes. Those nanoparticles, when administered into colorectal cancer, CT26 murine tumor model, when administered into mice, we noticed surface exposure of calreticulin and increased dendritic cell maturation, and this resulted in reduction of the tumor size. Another strategy is using immunogenic cell death whole tumor cell vaccine. Here, the dying tumor cells after treatment of immunogenic cell death inducer will be coupled to nanoparticles loaded with an adjuvant, and the whole tumor cells will be used as a vaccine. So, for example, melanoma cells B16F10 expressing ovalbumin as a model antigen were treated with mitoxantrone for 12 hours to induce immunogenic cell death and help release of DAMs. Then these cells were coupled chemically to nanoparticles. These nanoparticles are loaded with toll-like receptor agonist, CpG, as an adjuvant to help enhance the activation and maturation of dendritic cells. So in this strategy, mice were injected with cancer cells. Those cancer cells are pre-treated with mitoxantrone as immunogenic cell death inducer. And at the same time, the cancer cells are surface decorated with nanoparticles loaded with adjuvant to enhance the activity of dendritic cells. So after injection, the dendritic cells will uptake the antigens released by the immunogenically dying tumor cells and will be activated by the released DAMPs. And the matured dendritic cells will infiltrate, will prime first the T cells, and T cells will infiltrate the tumor and attack the cancer cells. In this strategy, vaccination of mice with mitoxantrone treated tumor cell coupled to CpG nanoparticle as adjuvant resulted in enhancement of the phagocytosis of cancer cells by dendritic cells, also enhanced the cross-presentation of the model antigen of albumin from melanoma cells, increased the maturation of dendritic cells as demonstrated by enhanced expression of the activation marker, for example, CD40 and CD86, also generated strong antigen-specific CD8 positive T cell, and protected all animals against tumor initiation. So the vaccination combined with immune checkpoint inhibitor resulted in complete tumor regression with about 80% regression in about 80% of colorectal cancer-bearing mice. Finally, how to test the immunogenic cell death induction power of your nanomedicine, both in vitro and in vivo. We can analyze the detection of DAMPs, calreticulin surface exposure, release of HMGB1, ATP, interferon-1. We can also test the maturation and activation of dendritic cells and the macrophages. And finally, we can analyze the infiltration and the cytotoxic function of T cells infiltrating into the tumor. So, for example, in this research study using oxaliplatin and the dihydroartemisinin nanoparticle to induce immunogenic cell death, the authors here analyzed the surface localization or exposure of calreticulin here using confocal microscopy. It can be also analyzed using flow cytometry. So we can notice increased expression of calreticulin on the surface of cancer cells after treatment with immunogenic cell death inducer compared to non-treated cancer cells. Also, we can quantify the amount released of HMGB1 into the conditioned media of cancer cells after treatment with the immunogenic cell death inducer. We can quantify the level of HMGB1 released in the media using ELISA, for example. Also, we can detect the apoptosis of cancer cells after treatment with immunogenic cell death inducer nanomedicine using very simple fluorescent tagged annexin V analyzed by flow cytometry to quantify the amount of apoptotic cells. We can also analyze the infiltrating dendritic cells and macrophages into the tumor. So after treatment with the immunogenic cell death inducing nanomedicine, we usually notice increase in the percent infiltrating CD11C positive cells, which is dendritic cells, and F480 positive macrophages. And also we can quantify the amount of matured dendritic cells by using flow cytometry using its activation marker CD80 or CD86. Finally, we can analyze the infiltration of cytotoxic T lymphocyte, CD8 positive T lymphocyte into the tumor. So after treatment with immunogenic cell death combined with immunotherapy, there was a significant increase in the infiltrating T cells into the tumor. We can also analyze the secretion of interferon gamma by T cells, or also we can analyze the percent of effector memory T cells into the tumor. Thank you.